viewers. So we are studying metabolism since last many weeks. So I thought that today you should check your concepts, correct? So I have chosen some multiple choice questions for you. So, and I hope that you all have revised the concepts in the last few weeks, correct? So we should start now. Okay, so we will start from the first question, which is from glycolysis. The most important and controlled step in the glycolysis is, and there are 4 options given. So if you have a quick revision or if you have revised, you can have an idea that it is the formation of fructose 1,6-bisphosphate. It is the reaction conducted by PFK1, or it is the reaction having high negative delta G, and we have discussed this in very detail. Okay. Second, activators of enzyme pyruvate kinase includes. So in the regulation of glycolysis video, I have made a chart for you, and through this chart you can easily say that yes, it is fructose 1,6-bisphosphate, which is the activator of enzyme pyruvate kinase, which actually does the feed forward activation, and insulin also in this case. Yes, we have not discussed the hormonal regulation of these enzymes, but insulin it is active under high blood glucose condition, and glycolysis is done only when your glucose concentration in the blood is high, correct? So hormone insulin and fructose 1,6-bisphosphates, these two are the activators of pyruvate kinase. The higher concentration of these two things say that yes, there is there is enough glucose in our blood and we should go for glycolysis, correct? Now the third question is, which of the following enzymes are regulated? Okay, and the options are glucokinase or hexokinase, aldolase, pyruvate kinase, A plus C and none. So in this question, pathway is not mentioned, like whether it is from glycolysis or PDH complex or from Krebs cycle, it is not mentioned. But if you have studied glycolysis, you can say that these all are enzymes of glycolysis. Okay. So when we were studying glycolysis, we studied that hexokinase and pyruvate kinase, these two are regulatory enzymes of glycolysis, correct? Coming to the next question, the enzyme having low Km and low Vmax for glucose is. See, whenever I say low Km, it means high affinity, correct? So low Km means high affinity. So among all these enzymes, we know that glucokinase is the liver enzyme and its affinity for glucose is low. So low Km, the enzyme having low Km and low Vmax for glucose is hexokinase, because it has high affinity for glucose. It can sense low concentration of glucose and go for glycolysis, correct? Next, which is inhibited by glucose 6-phosphate? From where this moiety comes from? It is the first intermediate of glycolysis, okay? And obviously it should be inhibited by either hexokinase or glucokinase or both, but glucokinase is a liver enzyme and it is not inhibited by glucose 6-phosphate, and we have discussed this in the glycolysis video. So the correct option is only hexokinase. See, do not confuse with the options A plus B or B plus C every time. These are not correct every time. So you need to be very clear with your concepts. These options are given to you to make you confused. But if your concepts are clear, no one can confuse you, correct? Next, glycolysis, and these 4 statements are about glycolysis. Let's read one by one. It doesn't require oxygen to generate energy. Is it true? Yes, it is true, because in the glycolysis 2 ATPs are directly formed. Next option, we need to study all the options. Requires oxygen to generate energy. Yes, but not 100%, because NADHs which are generated in glycolysis has to donate their electrons in the oxidative phosphorylation. Okay, but ATP, 2 ATPs are directly directly generating in glycolysis in anaerobic conditions. Glycolysis is inhibited by oxygen? No, it is not the case. And its rate is increased in the presence of oxygen? No. Glycolysis has no effect on the level of oxygen. So, the correct answer is the first one, that it doesn't require oxygen to generate energy. Although without oxygen it generates less ATP, but still it is generating ATP in absence of oxygen. So, the next question is, see, this is the question only, and I have made these charts, or I have selected these charts for you to make this question easy to understand to all of you. Okay. So, the question is starting from glucose 6-phosphate to 2 molecules of lactate, the net ATP produced is. This is a very tricky question, and I request to all of you to focus on every word of the question and then on to these pathways, correct? So, from the right picture, it is glycolysis from step 1 to step 4, and the left one is the glycolysis from step 5 to step 10, correct? Glycolysis is a 10-step pathway, and it is not possible for me to write every step in a single line. So, I have cut down the pictures into 2 parts, and here it is for your consideration, correct? So, the question is starting from glucose 6-phosphate. So, what we need to do is, we need to skip this step, correct? We have to start from glucose 6-phosphate, correct? So, if we are eliminating this step, means we are eliminating this ATP consumption, correct? So, let us count upon the ATP only from glucose 6-phosphate to pyruvate, correct? So, it is 1 ATP minus 1 ATP here, plus 2 ATP here, and plus 2 ATP here, means a total of 4 ATP in output and a total of 1 ATP as an input in the step 3. So, we get 3 ATP if we start from glucose 6-phosphate, okay? But this is not the end of this question, because the question asks about starting from glucose 6-phosphate to 2 molecules of lactate, the net ATP produced is, correct? So, we need to move ahead from pyruvate to lactate, and this is the reaction of LDH, which shows that 1 NADH is consumed and 1 NAD plus is produced. So, most of the students, what they do, that they will multiply NADH as per their equivalent to ATP, which is around 2.5 or 3, and then state the answer, correct? But this is not the case, because the question ends on lactate production. It does not say that starting from glucose 6-phosphate to 2 lactate, the net ATP produced in oxidative phosphorylation, correct? So, the net ATP from the glucose 6-phosphate to lactate is only 3, because none of the ATP generates from pyruvate to lactate. Because one more point is that the NADH, it is not generating in this step, rather it is oxidizing in this step. And second, this question does not say that in oxidative phosphorylation how many net APs you get from glucose 6-phosphate to anything, correct? So, number 3 is the correct option. Okay. So, this is how these days teachers confuse you with by asking questions in such a manner and by giving options in such a manner, and the only solution of all these problems is to make your concepts all clear, correct? Now, let us move towards the another question. Which of the following slows down TCA? And recently we studied that high energy content of the cell means high concentration of ATP and NADHs slow down tricarboxylic acid cycle, correct? It was a simple one. Now, all of the following cofactors are required in PDH complex except, if you remember the PDH complex, you might know that lipoic acid is required as by complex 2, and NAD plus and FAD both are required by complex 3. So, all these are required in the PDH complex. There is no exception given in the option, correct? Now, the quite tricky question, the last one. When having thymine deficiency, which enzymatic activity do you expect to be decreased? Okay. So, this question does not say that thymine is a cofactor of any enzyme. We need to think that out of these enzymes, which enzyme uses thymine as a cofactor? Is it pyruvate carboxylase? No. Is it malate dehydrogenase? No. Is it fumarase? No. Is it alpha ketoglutarate dehydrogenase? Yes, because it is similar to pyruvate dehydrogenase complex, and the complex 1 of PDH requires thymine as a cofactor in the form of TPP, thymine pyrophosphate, correct? So, the fourth is your correct option, correct? And it was long. I have not given any homework question to you. So, this is for you, and since I am putting various effort in making slides and study material for all of you, so I expect the answer in the comment section from all of you. Please write. Happy learning.